I'm Peter Carlton, an associate professor in the Graduate School of Biostudies. My research group studies meiosis, the special cell division that in animals creates sperm and egg cells. What makes meiosis special is that unlike a normal cell division, meiosis creates daughter cells that have half the diploid chromosome number. This reduction in chromosome number is critical because at fertilization, the genomes of the sperm and egg cells combine to create the diploid chromosome number. This means that most of our cells in our body contain 2 of each chromosome, one from our mothers and one from our fathers. In meiosis, these pairs of chromosomes must find each other, pair along their lengths, and recombine so that they can biorient and be segregated into opposite daughter cells. How chromosomes find each other in meiosis and manage to orient in opposite directions and divide properly has been the focus of my laboratory at Kyoto University for the past 12 years. We're interested in 3 main areas: how chromosomes find each other, how they recombine and exchange genetic information, and how they segregate correctly during the first meiotic division. We used the model organism Caenorhabditis elegans, a tiny nematode worm made up of only about 1,000 cells. C. elegans has several advantages. It has a generation time of only 3 days, so genetic experiments can proceed quickly. It's used around the world by thousands of researchers, so its biology is very well understood. Its body is small and transparent, so microscopy is easy. And almost half the cells in its body are germline cells undergoing meiosis, so it's very well suited to our research. Our laboratory uses a combination of approaches in our research, including genetics, biochemistry, microscopy and image analysis, and bioinformatics. Here, we have the equipment and facilities to support all these approaches. Each researcher has their own bench and microscope, which we use to manipulate and roughly examine C. elegans. More detailed examinations of cells and chromosomes require more advanced microscopes, which we have here. For example, this microscope is used to examine GFP fluorescence in worm bodies, while this one is for taking high-resolution, 3-dimensional images of cells. We also use this microscope with a microinjection apparatus to introduce DNA into worms for genetic engineering. With the CRISPR-Cas9 genome editing technique, we are able to make virtually any change we want to the worm's genome, then see the effect these changes have on chromosome dynamics during meiosis. One of our main questions is how chromosomes recombine or exchange genetic material during meiosis. To recombine, the DNA of a chromosome must first be broken. This is usually a dangerous thing that cells try to prevent, but in meiosis we create breaks on purpose in order to enable recombination. We would like to know how the number and position of DNA double-strand breaks is regulated during meiosis. We would like to know how the number of DNA breaks is controlled. There can't be too many breaks, since this could lead to DNA damage and mutation. But there also can't be too few breaks, since each chromosome needs to get at least 1 break to ensure that recombination happens. We have found a feedback mechanism in meiosis that turns off the proteins that promote DNA breaks when the number of breaks gets too high. We're currently studying how this feedback works by making changes in the proteins that carry it out. One recent exciting development in biology has been the ability to predict protein structures using artificial intelligence methods like AlphaFold, and we are using these methods to make better predictions about what parts of the proteins are involved in DNA breakage and how these proteins interact. Our lab has had students and other researchers from Japan and all over the world, and we all use English in our daily scientific discussions and weekly lab meetings. Many students have found their scientific English improves dramatically after spending time with us. I hope you'll contact us if you have any questions.